Yeah, it's really exciting to be at the first ever of these sessions. So I'm originally from Sydney. I did my undergraduate at UNSW, and then I moved to Adelaide in 2007 to do my PhD. And I actually did it within the myeloma research lab, but was working on a project looking at chronic myeloid leukemia and looking at side effects of those drugs that are used to commonly treat myeloid leukemia. So drugs like imatinib or Gleevec, you might have heard of. And then because I was based in the myeloma lab, that kind of got my interest in looking at myeloma cancer. And from then, when I finished my PhD, I started looking at myeloma. And I've been in that lab ever since. As Nikki said, I'm starting from late last year, I've become the co-lead of that lab. So we've got about 25 people in that group.
Wonderful. Thank you, Kate. So what sparked your interest in cancer research? Before we even get to the myeloma piece, why did you start there, and why did you want to look at cancer research?
So I think in undergrad, I really got interested in hearing about how cancer cells interact with the normal healthy cells in a particular tissue, so particularly in bone, and how cancer cells kind of corrupt the normal cells and the normal microenvironment that the cancer exists in to support its growth and to enable it to kind of grow out of control. And that interaction, how the cancer cells interact with those normal cells, really interested me. So I did an honours degree in Pam Russell's lab at the Prince of Wales Hospital on prostate cancer and looking at prostate cancer, particularly in bone metastasis. And that really got me interested again in those interactions. And from there, it really made sense to make that switch to multiple myeloma, which is also a bone cancer. So it was really kind of a nerdy interest in how cancer works that really got me interested in that area.
So then moving on to myeloma, multiple myeloma, I've been fortunate to see a couple of your presentations before, and you best describe this as the bones in someone's body looking like a slice of Swiss cheese, if you like, where it's all gone wholly, and the bones start to deteriorate. And that really resonated with me, because not having a scientific background, it made a lot of sense. Do you want to just talk a bit more about multiple myeloma and how it occurs in the body, and also what are the side effects of it?
Yeah, so myeloma is it's typically cancer of the elderly, so it affects people at about age 70 on average, but it can be a lot younger. And with our aging population in Australia, we actually have one of the highest incidences of myeloma in the world. And it's a disease of a white blood cell called a plasma cell. It's normally involved in fighting infection, but in myeloma, those cells start growing out of control in the bone marrow. And that causes all sorts of problems. So that Swiss cheese bone that we see, which is hugely debilitating for patients. So they get literally, the cancer causes the normal bone cells to eat holes in the bone, which causes fracture and a lot of pain and other issues. And we also see other things like you get kidney dysfunctions and also anemia, just because of and suppression of the normal immune system, just because of the effects on the normal bone marrow cells. So it can be really debilitating. And we currently don't have any cures for this cancer. So unfortunately, everyone who gets this disease will eventually succumb to the disease.
Okay. And when you talk about it being more of a disease of the elderly, how does it start? Is it just over time in the deterioration of human body, or are other factors at play at getting myeloma?
So it's something it doesn't really seem to be associated that much with environmental factors or with family history. There is a small family history associated with it. If you've got relatives with it, you can have a slight increased risk, but it tends to more just be a random thing. So the thing with plasma cells is that the way that they make antibodies to fight infection is that they throw bits of DNA around in the cell to make those antibodies. And part of that process of throwing DNA around can cause errors, which cause mutations that can lead to the cancer. So it tends to just be a kind of a random process that leads to the development of these abnormal plasma cells that then go on to become cancer.
Okay, thank you. And then you talk about there is no cure, sadly, from it at the moment. Why is that? Have we not done enough research, or is it just very tricky to find a way to treat myeloma?
Yeah, so I guess being a bone marrow cancer, for starters, you can't use surgery to cure it. You have to rely on drugs. And so that's one of the problems that we have. And cancer itself is extremely heterogeneous. So there's lots of different mutations that can cause it. So we can't come up with one specific targeted therapy that will cure everyone. And bone marrow cancers tend to be pretty sneaky, and those cells can hide in the bone marrow. They're relatively protected from chemotherapy and other drugs. And so that can lead to the development of resistance, and then you get relapse, where those cancer cells, any cancer cells that are remaining after treatment can relapse.
Yeah, okay, thank you. And one of the questions we just had from the audience is, what's the incidence? So as you age, how likely in Australia are you likely to get multiple myeloma?
So we have about 2,000 people every year in Australia that are diagnosed with myeloma. So it's the second most common blood cancer after lymphoma. So it's, it's not as common as, say, breast cancer, prostate cancer, but it is quite common in the realm of blood cancers. It is quite a common one.
Yeah, lovely. Thank you. Okay, well, let's just move on to talk about your research. We'll be talking about two pieces of research, one that you have just finished up and then your newer focus. So in regards to the one that you've just been doing, do you want to talk about what your area of focus is there and also what results you've seen coming up to?
Yeah, so with the project that we've just finished up, it really started from talking to patients about their cancer journey and what are the things that most impact them. And one of the things that is a huge problem for myeloma patients is actually the side effects of the drugs that they go on. So one of the most common drugs that we use is something called bortezomib, and that's most commonly used up front in the treatment of myeloma in combination with some other drugs. And in up to a third of patients that are treated with bortezomib, we see something called peripheral neuropathy, which is basically it's a side effect of the drug where it starts attacking the nerve fibers that supply your hands and feet. And so patients get numbness in their hands and feet, they get tingling, burning pain, and this can really, really affect their quality of life. So someone who might not have had huge side effects from the cancer are then finding that the treatments that are on make their quality of life so much worse than what they were like when they were first diagnosed. And that's a huge issue for patients because it makes that choice to go on that treatment really difficult if they know that it's going to cause these side effects. And it can mean that we have to reduce the dose of the drug that we use, which means that we're not going to get as good a control of their cancer. So we were looking at ways of decreasing the side effects of this drug, and we discovered another drug that we can use in combination with bortezomib. That basically means we can reduce the dose of the bortezomib that we use. So that means that we can stop those side effects, or at least decrease those side effects, and then potentially enable patients to get a better control of their cancer and remain relapse-free for longer. So when we don't like to talk about a cure for myeloma with these drugs, but at least that can get that cancer under control for as long as possible before they have to go on a different treatment.
Yeah, look, it's something we hear often, isn't it? That unfortunately the drugs that are used for the different types of cancer are so severe it has knock-on effects for patients going through that treatment as well. So how did you discover combining these drugs? How did you know that this combination would have a better side effect as well as a good result for patients with myeloma?
Yeah, so it was kind of fortuitous because we were looking at this new drug that we found. We were looking at it in a different context in myeloma biology. So we thought it might be useful to decrease the spread of the cancer cells. So we were looking at it in that context. And in some early studies we did there, we found that if we used this new drug in combination with bortezomib, we got this really dramatic effect of combining the two drugs together, way more than we got with each drug individually. And so we started looking into how that was occurring and really found this amazing effect that this new drug that we have has on targeting bortezomib to the cancer cells. So it enables the drug to get to the cancer cells much more effectively. And so that means we can reduce the dose of the bortezomib that we need to use, and thereby decreasing those side effects. So we've got some beautiful data that were produced with this grant that we got from you guys a couple of years ago. We have beautiful data in mice showing that we can use a much lower dose of drug to treat the cancer, and also that at that lower dose we get basically none of these peripheral neuropathy side effects that we see with a high dose of the drug.
Well, that's I mean, that's fantastic, isn't it? That's just what we want. So where are you with that? So it works in mice models. Where are we to take that to humans and actually have a positive impact in their treatment plan?
Yeah, so we hope, so we've got beautiful data in mice. And so with that, we're hoping that we'll be able to take this drug to phase 1 and 2 clinical trial. We haven't used this particular drug in humans before, so we'd have to go through those initial safety studies, but we've used similar drugs before, and there've been other drugs on the market that use a similar sort of, they're targeting a similar thing. They're not being used in the same sort of context, but they're targeting the same thing. So we know that those drugs are safe, so we're hoping that this one will be safe, and certainly in mice we don't see any issues with using this drug. So the next stage would be doing those safety studies and then taking it to clinical trial to see if we can use it to decrease side effects in patients.
Lovely, thank you. And then what's the treatment plan for someone that does have myeloma? How do they get this intravenously? How would that work for those patients?
Yeah, so typically these patients get they get 3 drugs to start off with to treat their myeloma, one of which is bortezomib, and this would mean that we could add in this other drug that we found, add that in, so they get 4 drugs in combination. And yeah, they go into hospital and get this the bortezomib intravenously, and we just add in this other drug into that infusion so that we could increase the delivery of the bortezomib to the cancer.
Wonderful, thank you. And then, you know, your hope on this project for clinical trial, how far away, if all the safety pieces are done well and approved, how far away do you think it will be for us to get to a clinical trial with this combination of drugs?
Yeah, so obviously you need to go through ethics and get funding for those clinical trials, which is always a couple of hurdles that you've got to go through. But I think potentially in the next couple of years we could see, you know, a good strong clinical trial being started to test these drugs out.
Wonderful. I mean, that's very exciting. So thank you, and well done to your team. So let's move on to the next piece of your work then. Do you want to talk a bit about what your focus is now?
Yeah, so when we so I mentioned bortezomib, that is, you know, the most commonly used upfront treatment, but we have a real range of different treatments that we can now use for myeloma. Over the last 20 years, we've seen a real variety of different drugs that are being used for myeloma. So in the 90s, the survival for a myeloma patient would only be around 2 years, and now the average patient will live 5, 6, 7 years. So there've been huge improvements, but we don't have a good way of knowing which patient is going to respond to which treatment. And as I was saying before, resistance or the acquiring of resistance and then relapse of the cancer is very common to these therapies. And so patients often feel like they're a bit of a guinea pig because they're getting tested out on different drugs. They go on one drug and then it might work for a little bit, and they start to relapse and get resistance and relapse, and they put on a different drug, and they can go through these multiple cycles of different therapies before they can find one that's really going to work well. So what I really want to do is to find something that we can use to identify who's going to respond better to which particular drug. And also look at ways to decrease the resistance that we see in some patients. So we see up to, you know, a quarter of patients will have intrinsic resistance where they don't respond to, for instance, bortezomib upfront. And so if we can work out who those patients are going to be, and then ways of improving the effectiveness of the bortezomib for those resistant patients, then we can really improve outcomes potentially.
Yeah, that'll be great. How do you do that? How do you look at the markers or look at the impact of these different drugs for these individuals, and you know, get enough data sets to decide that this patient's showing X, whilst this patient could respond to Y type of drug as well?
Yeah, so we started with using so data sets that are available, looking at all the genes that are expressed in the cancer cells from multiple different patients. And we have these data sets where we know the outcomes for those patients in terms of did they do well or not following treatment. And what I did was I kind of mined that data to look for things that were cell surface markers, so things that we could detect in our normal clinical testing that we do on the cancer cells, that could potentially use as a marker for their subsequent outcomes. And so we identified one particular protein called CCR1, which is high in about half of patients. And we know that patients who have high levels of CCR1 are the ones that tend to do very poorly. And so that kind of led to us looking at the underlying biology of CCR1, what is it doing in those cancer cells, and led us to look at whether it was associated with a poorer response to particular drugs. And that's something that we're looking at in this current grant, is whether patients who are on particular drugs will do better or worse, depending on their expression of CCR1. And so then if we could test them at the start and say, okay, you have high CCR1, we'd be better off putting you on this drug because you're going to be doing better from that.
Yeah, I mean, when you talk about it like that, just to think that not only have you been diagnosed with this incurable cancer, but then we don't even know in 2022 what drug would be better or more effective on you. I mean, it really is a bit of a guinea pig, to coin a phrase, isn't it, for those poor patients going through this treatment at the moment?
Yeah, that's right. And it's really stressful for them because often they might have gone through one round of therapy that worked quite well. But then now they've developed resistance and relapse, and then they have to go through that whole stressful process again. And all I hear from patients all the time is how much they dread getting those blood tests. They get blood tests every 3 months to see whether their cancer is coming back. And they just dread that hearing that number and knowing that they have relapse, and then they have to go through this whole cycle again of, will they respond to this new drug? Are they going to get side effects from that treatment? And what's their ultimate outcome going to be? Is this drug actually going to work?
Yeah. So what's your hope? Because obviously there is no cure at the moment, but if we can find a better treatment plan for the patients, is it that they can live longer, or they live a more pain-free existence for the time that they are with us?
I mean, I think it's really about quality of life. I think that is really the most important thing for everyone, really. If you don't want to be getting treatment for your cancer and knowing that your cancer is under control, but not being able to leave the house because you've got in too much pain. So I think that from the perspective of most of the patients I talk to, that quality of life issue is really the most important. So if we could come up with, I mean, ideally both, something that's going to be effective at keeping the cancer under control, but also have minimal side effects for those patients who are living longer, but then they have to have these side effects for longer as well. So if we can try and minimize that and improve just the quality of life of patients, that would be ideal.
And what's the average lifespan if someone does get multiple myeloma? What are they looking at in terms of lifespan for the next few years?
So we know that about a quarter of patients will only live a couple of years from diagnosis, and those are the ones with the most aggressive cancer, or the ones that respond the worst to the therapy, so that they don't get that good control of their cancer. The average patient would live for 5 years, and then I know patients who have lived 20 years after initially being diagnosed. With all the current therapies they're coming up with, we have lots of new options when patients do relapse. We've got new things that we can try them on. But yeah, they do end up being a bit of a guinea pig, testing them out on new things or being put on clinical trials. So, but in terms of outlook, it's a lot better than it was even 10 years ago.
Right, thank you. And how do you know that you have myeloma? Do you get any outward symptoms as an individual that you'd be able to identify, or is it purely from blood work and tests in the lab?
Often the kind of symptoms that people have are quite non-specific. They might be a bit tired, might have a bit of pain. And because of that, it often can take quite a while before someone's diagnosed. I was talking to a patient the other day who was diagnosed just in January, and he had some issues, a bit of back pain, a bit of hip pain way back in January of last year. And his doctors kept on fobbing him off as being, you're just older, you're getting a bit of arthritis, it's back pain, just have some analgesia, you'll be okay. And then, it got worse and worse to the point where he had to present to hospital, and then they ran the definitive blood test that found that he did have myeloma. So it's something that either it is these kind of non-specific symptoms, or it is just something that can just pop up on a blood test where, someone finds they've got, the doctor might notice they have high protein in their blood, and then that kind of follows along and they work out that that's because of the cancer cells. So it's a tricky one to diagnose, and I think it goes underdiagnosed a lot, which means that patients are often a bit more advanced when they are diagnosed with myeloma.
Yeah, okay, thank you. Just a question about that and being diagnosed. So when you do find out you have multiple myeloma, and you talk about a concoction of drugs according to responsiveness. In a couple of other examples of cancer types, we've found that different drugs, for example, arthritis drugs, are now being used to treat pancreatic cancer. You just talked about using a combination drug that's already exists as well. Do you think there's the future there about exploring current existing drugs so that you don't have to go through that whole approval process for new drugs that come onto the market?
Yeah, so with this current study, this new study that we're starting with you guys, that was funded last year, we're actually using a drug there that has been used for rheumatoid arthritis previously. So it's actually something that that drug's gone to clinical trial, and so we know it's safe. We know that it's going to be well tolerated by the patients, and now we're going to see whether in this new indication whether we can use that to, in this case, decrease drug resistance for myeloma patients. So, yeah, absolutely, and it's drug companies often put so much money into developing these drugs and putting them through all the initial studies, and then they might not work out for a particular indication. So if we can repurpose those drugs for a new indication, that's fantastic, and we'll save a lot of money in the long run as well.
Yeah, no, absolutely. Thank you. Just a reminder for anyone just to pop any additional questions you may have. Thanks for those that have already done that. So we also talk with other researchers that they find that treatments or markers for their type of cancer can also be used for different types of cancer. And obviously there's multiple blood cancers. Do you find there's a crossover of treatment plans or drugs across your different blood cancers per se?
So with the therapy that we're using to decrease side effects in myeloma, so with using it in combination with bortezomib to decrease that peripheral neuropathy that we see in patients, that drug that we've discovered could actually have potential to be used for any kind of cancer, because there's a lot of drugs that are delivered intravenously and travel through the bloodstream in the same way as bortezomib is. And the drug that we found, we haven't tested it in other drug combinations yet for other cancers, but it has the potential that it could decrease side effects for a lot of other cancer drugs. And there's lots of other drugs that are used that have issues with toxicity. So we think it's really exciting, and being able to test this in the myeloma space to start off with, but then potentially get other collaborators on board to test it with other cancers as well, will be really exciting in the future.
Lovely, thank you. Question about side effects. Often we hear, you talk about patients getting numb hands, etc. I've got a question here about fertility. I mean, that may not be relevant because these patients tend to be older, but are there any other significant side effects that are had from treatments at the moment for multiple myeloma patients?
Yeah, so one of the other things that we're looking at actually is gastrointestinal side effects. So often people get issues with their gut, so they have diarrhea or constipation or other issues that are also really, really big issues with patients' quality of life as well. And that's seen really commonly with bortezomib, but also with a whole lot of chemotherapy drugs. So we've actually been looking, we collaborate with someone else who's at SAMRI, who is an expert in that gut health area, and we've been looking at that in our mouse models as well, looking at whether or not we can decrease that side effect. So being able to stop, the direct toxic effects on the gut to kind of improve those sorts of side effects as well.
Yeah, thank you. A question I have actually, because we've funded multiple myeloma researchers at SAMRI. Why SAMRI or the University of South Australia? Is there a specific focus there for blood cancers?
Yeah, so we're part of the cancer theme at SAMRI, which, although it covers all cancers in theory, there's a lot of blood cancer groups. So we work with a lot of researchers on our same floor as us on SAMRI that work on leukemia, various different kinds of leukemias, and obviously us with the myeloma group. So there is a really strong focus at SAMRI on the blood cancers, and makes it a fantastic place to work. And we have a really excellent biobank as well that collects specimens from patients with cancers, particularly blood cancers, which means that we can do the sorts of studies on biomarkers and things like that isn't quite as easy when you don't have access to those patient samples. So at SAMRI, we're right next door to the Royal Adelaide Hospital, and we have great linkages with the clinic there, which means that we can get these samples and be able to do this test. Hopefully then feed it back into the clinic when we take things to clinical trial.
Yeah, absolutely. Thank you. Question here about CAR T cell therapy. It's a piece of research we've funded in the past down in Melbourne. Are any of your patients you're treating receiving that, or for example, is immunotherapy a positive response to those with multiple myeloma as well?
Yeah, so CAR T cell therapy is definitely being looked at in myeloma. Those studies are mainly coming out of Victoria. And we do have some patients that are being enrolled at the Royal Adelaide Hospital on those CAR T cell studies. The problem we see in myeloma is that the immune system tends to get dampened down, so it doesn't have as strong an anti-tumor response. And so using something like CAR T cells, where you can target those immune cells to the cancer more specifically, is really promising in terms of having a direct way of attacking the cancer cells. But yeah, those studies are quite in the kind of early phases in myeloma, but it is a really exciting area of research, and hopefully in the future that will be a really excellent way of treating the disease if we can get the side effects of the CAR T cell therapy under control, because it can be quite a difficult therapy to go through. It really can knock people about, and so you have to kind of pick your strongest patients who might be able to just withstand that in order to potentially get a cure for their cancer.
Yeah, thank you. Just moving back and talking about detection, you mentioned how there's just very typical symptoms of someone being sick. Obviously, I think in most cancer cases, if you can detect it earlier, you've got a likelihood of living longer. Is that the case in multiple myeloma as well?
Yeah, so with myeloma, it's almost universally preceded by a precancerous condition. So there's two different ones. One called MGUS, which is seen in about 1 in 30 people over the age of 50, where they have very low levels of these cancerous cells present in their bone marrow, but they don't start growing out of control. And then there's another condition called smoldering myeloma, where they have a bit more of those cancer cells, but they're not causing any of those debilitating symptoms. So they're eating the holes in the bone or the kidney dysfunction or anything like that. And under both those conditions, you have a chance of going on to develop the active myeloma. But at the moment, we can't predict who will be the ones that go on to develop myeloma. And there's lots of work being done in the US, especially on looking at earlier treatment for those patients, especially if we think that they have a higher risk of developing myeloma. Maybe we can start treating them earlier and then improve survival outcomes. And those studies have been really promising in terms of showing that earlier treatment does, in fact, delay them developing the cancer, myeloma, delay those horrible symptoms, and also in the end improve their overall survival. And so one of my colleagues, Melissa Cantley, who you've also funded, is looking at developing a blood test, so blood biomarkers that we can use to predict who are those patients with those preceding conditions, so smoldering myeloma, which ones are more likely to develop myeloma, so we can then specifically target those patients with treatment, prevent them from cancer.
Thank you. So just a question about funding. Obviously, we've helped support you. The Snowdon Foundation has helped support you as well. Obviously, this funding is critical because there is no cure, and it's a bit potluck at the moment for patients when they go through the treatment. What type of funding comes from the government? How well funded is this type of research in Australia?
Yeah, so we're reliant, obviously, as everyone else is, on the NHMRC, which is our biggest funder. And over the last several years, our chances of getting funded through the NHMRC has gone down and down and down. So now it's less than 10% of those grants that we apply for get funded. And it's becoming particularly difficult for early career researchers and mid-career researchers to kind of get their foot in the door with those funding schemes through the major funders. So we rely a lot on charities and things like that for funding. And obviously with COVID and all the issues that charities have had with fundraising, that those are getting more and more competitive as well. So it is extremely challenging at the moment for getting any support for this sort of research. It means that we're actually losing a lot of our early career researchers who might have just finished their PhD and are brilliant people, but can't get the support they need to continue with their work. So it's really a huge issue, and it's something that is it's really sad actually to see all these fantastic junior researchers that have the potential to lead to those new ideas and those new treatments, and we're really losing them out of the system. So yeah, it's a challenge for sure.
Yeah, absolutely. And you talked about going to clinical trials, which ideally is where we all want to be. What are the costs associated of getting there, just to give people an idea of just how expensive it is?
Yeah, we're talking about hundreds of thousands of dollars to run even a relatively small clinical trial with all of the things that you need to put in place to get that to happen. So if you're working with, a drug that's already coming from a pharmaceutical company, obviously that pathway is a lot easier because hopefully they'll be happy to help support those trials. And you can also get co-funding. There's some government funding programs that do co-funding. So if you can get some support from industry, then you can get some funding from the government to do the clinical trial as well. But if you're coming up with a brand new drug, kind of at the grassroots level, you're trying to get that funding can be quite challenging, for sure.
Yeah, it's crazy when you think about it, because you're actually being more innovative, coming up with new ideas, and then that actually makes your funding harder, doesn't it?
Yeah.
So moving forward, obviously we're funding you just to do this second piece of work. What are your hopes for the future? You've talked about getting the right drug combination, looking at the markers to make sure we give the right combination to the right patients at the right time. Fast forward 5 years, 10 years, where do you want to see your research? What do you want to have achieved, hopefully?
Yeah, so I mean, ideally what we want to see is if a new patient comes into the clinic, you've just been diagnosed with myeloma, you turn up at your doctor, that they can add this test, this looking at the CCR1 into the normal panel that we look at when we kind of assessing a new patient. And if they were put in that high CCR1 group, we'd be able to say, okay, you're going to do better on this drug. And potentially, if we give it in combination with an inhibitor of CCR1, you might be able to do even better. So just being able to kind of personalize that therapy a bit more so we can pick the best person to get the best drug combinations, and to do that right at the start, not to have to kind of use them as guinea pigs to test multiple things before we find the right fit.
And in the long term, I mean, do you see there being a cure for multiple myeloma? Is that possible?
Well, I mean, with things like CAR T cells and that sort of thing, I think that potential is kind of there. That we're finding that patients who undergo this, very early stage research, they are seeming to have no detectable cancer left, and time will tell whether or not there is those few residual cells that are going to kind of sneak up later. So I think that's showing the most promise in terms of a curative therapy. It's now just looking at whether we can make that therapy as low impact as possible, so we can decrease the side effects of the therapy to make it so it's applicable to more patients.
Yeah, great, thank you. So we talk about the challenges of funding, etc. But why do you do what you do? What's rewarding? Why do you enjoy it?
Well, I mean, now that I'm the co-lead of the lab, one of the things that I'm loving the most is mentoring of the early career researchers, the up and coming researchers in the lab, and supporting students, PhD students, supervising PhD students. I find that incredibly rewarding, trying to help them to go through the ups and downs of science, which there are lots of ups and downs, and supporting them to do the best that they can in what they're doing is probably the most rewarding thing that I'm doing.
Great. And I mean, you talked about the challenge of funding, particularly for the ECRs coming through. Are you still seeing a pipeline of researchers that want to join your team and be part of that, or is that becoming progressively more harder as well?
Yeah, I mean, we are definitely see a lot of we're seeing a lot of students coming through. We get a lot of PhD students coming through, but being able to hold on to those promising students is tricky, because they're just seeing that there's not as much of a future in research when they see that the funding situation is so difficult. But we're really lucky to be a relatively well-supported lab at the moment. So we are being able to hold on to those really promising new students that we have coming through. We're being able to give them support to start their own projects in the group, and to kind of follow up on their own areas that they're interested in, which has been it's been fantastic to be able to kind of nurture those upcoming young researchers.
Oh, that's great. Thank you. Sorry, I seem to have a bit of a lag here. It's a long way between Sydney and South Australia, but we'll we'll get there. So, you're talking about the future and what you want to achieve. We have a wonderful team of people here, subcommittee members, observers. What would you impart to them? We're all here to support you and the ECRs. What do you need from us, and how can we help really progress what you're trying to do?
That's a good question. I mean, I think that the support that Cure Cancer has always given us as early career researchers, those opportunities to attend those research symposiums, to do presentations, to connect with other people, communicate our results, has always been amazing. And having those sort of career development sort of areas that you guys have always been so fantastic at. I think that you guys do that so well in terms of just giving people opportunities, helping us to build our CVs as early career researchers has been fantastic. Yeah, I'm not sure what else I can ask of you really in terms of what else you can give us. It's been fantastic. Yeah, I've been so lucky to have been given 3 opportunities to have funding from you guys, and it's been absolutely amazing in terms of enabling me to pursue the areas of research that I've been really interested in, and to build my group, and to now lead the lab as a whole. So yeah, it's been amazing.
Thank you. And then, I mean, really, just from your career perspective, we talked about the cancer research, but where do you want to be? Where do you see your career going over the next 5 or 10 years?
Yeah, well, as a kind of the new head of the group, I think that I'm going to be continuing to expand that group, to support the early career researchers in that group, and to continue the research. But really what I want to see is that the research that we're doing, that kind of new discovery research, we want to see it go from those initial findings in the lab all the way through to clinical impact. And that's something that we haven't quite got to yet. And that's the next step, I think, for all the work that we're doing, is to get this into the clinic, to get those biomarkers being used for patients, and to get those therapies out there. Yeah, see it really helping patients.
Thank you. And what about the opportunity for international collaboration? Obviously, we know they do a lot in the US on CAR T cell therapy. Where do you see that going in the next 3 to 5 years as well?
Yeah, so we've got collaborators all over the world that we work on with these projects. We collaborate with people in Germany, in the US, in the UK, and in the myeloma space especially. It is quite a nice, supportive, collaborative environment. There's this real feeling of working together to try and solve the problem, rather than being competing against each other. So it is a really nice space to work in. Obviously, we've been a bit limited with COVID in terms of connecting with people and being able to meet in person and going to travel for conferences and things like that. But we've been able to use Zoom to maintain those collaborations as much as we can. So yeah.
Well, thank you, Kate. Thank you for your time. Just to recap, I'm just looking at what you've mentioned today. We've talked about how multiple myeloma has one of the highest incidence in Australia due to our old aging population. We see about 2,000 people per year diagnosed with it, and it's the second most common blood cancer in Australia as well. And unfortunately, at the moment, a third of those patients have negative side effects from the treatment plans as well. So, talking about your aims, being able to find the right type of drug combination, and also understanding those markers within individuals going through that treatment, to make sure that it's less of a potluck and more of a controlled way of giving it to these patients too. And then from my notes, I also see, obviously we're looking at CAR T cell therapy is a big opportunity in the years coming up as well. So something that we'll definitely look forward to as well. Thank you for your time. We really do appreciate it. I know you'll be here for our World's Best BYO in a couple of months, and we can talk to you again. But for now, thank you for sparing this 45 minutes. It's been an incredible session to learn about the wonderful work you and your team are doing, and we very much appreciate your hard work. And we look forward to hearing your impending clinical trial when that comes in the next few years. So thank you, Kate, for your time. Thank you, everyone else, for joining us. What I think has been a very good session, and we look forward to the next session in next week. So until then, thank you, and goodbye.
Thanks, everyone.
Bye-bye.